Hello, everyone. Today we are discussing lecture number 4. This is part of module number 4. And today is one of my favorite topics. And I like anything related to microbiology, of course, but today is one of my favorite topics. Is about virology. This is what I do in the laboratory, research that I do working with viral sexually transmitted infections. So it is really hard to really talk about viruses in one lecture, only one lecture in the semester. So today I will give you just the basic tool to understand how viruses replicate, how they produce infections, how we can look at them in the laboratory and detect the presence of these viruses. And then at the end, we will talk about other acellular infectious agents like viroids and prions that are not viruses. However, when you look at any virology book, these acellular agents are still included in these books. Since there is not a particular place where we can place them, where we can classify them in terms of microbial organisms. So this is a slide that I showed you at the very beginning of this semester, where we try to organize, classify microbial world. And remember that to your left, you're seeing living organisms. They are made of cells. And you have the three main domains there: bacteria, archaea, and eukarya. And to the left, we have what we call acellular infectious agents. None of these acellular infectious agents are made of cells. And for that reason, it's hard to classify them within living organisms. And in these acellular infectious agents, we have viruses, viroids, prions. We will talk about each of them today. And we're going to start with viruses. When we talk about viruses, we are talking about obligate intracellular parasites. These are particles that contain a genetic material that is being protected by a coat. And these viruses on the external coat, they have what we call spikes that guarantee that that virus will be able to interact with a particular receptor that could be found in a particular tissue or particular cell. Now, if the virus is outside, the virus is just an inert particle. They don't have their own metabolism. They cannot replicate on their own. They don't have motility. They're just basically out there hoping that at some point they can find that cell that they can infect, that they can attach to, enter into that cell. And then now they will hijack the replication machinery of that cell and make that cell produce replicates of this virus. So most viruses, when you look at in terms of diversity of viruses, in terms of shape and size, we find a huge diversity of viruses. And that picture there is kind of showing you that we are able to find viruses that are extremely small, like the case of poliovirus there in that picture. There is only 30 nanometers right here. Or there are viruses that are so large that we can actually look at them under the light microscope. This here that is so small, for that we require an electron microscope to be able to look at those structures. In terms of size, the largest virus that has been identified is this virus that we call picovirus. It was first described in 2014, and it's a genus of giant viruses. They're huge. And these are viruses that infect protozoa. The discovery of this particular group of virus was done, I said, back in 2014 in Siberia, in Russia. And this is a virus that can measure about 1.5 micrometers long and 0.5 micrometer diameter. That is a huge viral particle, almost as big as a bacteria, let's say. And because they're so big, of course, they cannot infect bacteria. They cannot feed, into that bacteria. Instead, they're going to find eukaryotic cells that are larger, like protozoa, in which they can attach, enter, and replicate. And then on the other end, we have viruses that can be so small, like, for example, the case of porcine circovirus. This is a virus that is only 17 nanometers. Has a structure, the diameter of the structure is only 17 nanometers. So let's take a look at the structure of viruses. And the first concept that I want to introduce here is the concept of virion. Virion is a word that we use in virology to describe infectious viral particles. What I mean by that is that that viral particle is intact, is not missing any structure, and then because it's intact and everything is there in that viral structure, then that virus is going to be capable to infect and produce viral progeny. Now, sometimes viruses may lose, essential structure and become what we call defective viruses. These defective viruses are viruses that have loose, maybe the envelope on the outside, viruses that, empty capsids that didn't insert the genome inside of the viral particle, and those, of course, will not be able to produce infection. So those would be inactive. Now, there are two main categories of viruses based on the basic structure of the virus. And this applies mostly to animal viruses, what I'm going to explain to you here right now. We have what we call non-envelope viruses that also receive the name of naked viruses. And we have the envelope viruses. Let's take a look at the difference between these two main types of viruses. When we look at a naked virus is a virus that is made of a genome, that is a nucleic acid right there in green color, that is being coded by this structure in red right here, that is called the capsid. Now, when you have that capsid covering the nucleic acid, and remember in the case of viruses, the genome that is made of nucleic acid could be of different type of nucleic acid. It could be sometimes DNA, or it could be sometimes RNA. The DNA could be single-stranded DNA, or in some other viruses, double-stranded DNA. And in the case of RNA, it could be also single-stranded RNA. And for those, it could be positive or negative. And then we also have double-stranded RNA genome in some viruses. So that nucleic acid could be, different kinds. But when we talk about a naked virus, we are talking about that nucleic acid that is being encapsulated, coded by this structure that we call capsid. The function of that capsid is to protect that genetic material, because in the outside of the cell, in the extracellular medium, there could be enzymes that could degrade, that could destroy that nucleic acid. So that capsid is going to protect that nucleic acid. And in addition, that capsid is going to have on the outside a structure that we call the spikes. And these spikes are the ligands, the structures that will be in charge of finding a receptor on the surface of the cell that they can infect. And when that spike finds that specific receptor to which they can bind, then now the virus is able to attach, and then after that will enter the cell and start the replication there. This whole structure that combines the capsid, that protein code, and the nucleic acid, the genome, we call that the nucleocapsid. Now, in the case of envelope viruses, you still have that nucleocapsid loop here inside, but now that nucleocapsid is going to be coated on top of that by an envelope that the virus acquires from the cell. And in that envelope, the virus inserts the spikes that again will be in charge of trying to find that receptor on the surface of the cell that they can infect, interact with that receptor specifically, and then now start the process of attachment and entry into the cell to replicate inside of that cell. Viruses also can be classified based on the structure of that nucleocapsid. Viruses can be classified in different groups. We have these icosahedral shapes that you see right here, or they could adopt an helical structure like you see here. And here is a icosahedral. So this is a diagram here. This is the real virus using electron microscopy. You can see that shape there. And then finally, there are viruses that you cannot classify into icosahedral or helical because they combine different structures. Like for example, this bacteriophage that you see right here. And in this case, we're going to call these viruses complex viruses. We're going to start now talking about bacteriophages, and bacteriophages are viruses that infect bacteria. We also call them just phages. And after this, we will move to animal viruses. And at the very end, we will talk about other cellular infectious agents like viroids and prions. So let's start with bacteriophages or phages. The first thing that you need to know about bacteriophages, these viruses that infect only bacteria, is that there are three general types of bacteriophages. We have lytic phages, we have temperate phages, and we have filamentous phages. In all three cases of viruses, they go through a 5-step process in order to replicate inside a bacteria. The first thing that they need to do is they need to attach to the surface of that bacteria. And for them to attach to that bacteria, that virus needs to have a spike that is able to interact with a molecule on the surface of that bacteria, and that molecule will become the receptor for that virus. Some bacteria may have that receptor for that particular virus, others not. So that will indicate, the tropism of that virus that may be able to infect a particular species of bacteria that has the receptor for them, but maybe not others, not other bacteria that lack that receptor on the surface. The next step will be entry. So that virus needs to get the viral genome into that cell in order to start in the next step the synthesis replicate that genome, but also start the synthesis of viral proteins. When those viral proteins are produced, when the genome has been replicated, then now new viral particles can be assembled. And at the end, the virus needs to find a strategy to get out of that cell and be released. And you could imagine that if we're talking about lytic phages, the strategy that they will find to be released is lysing that cell, destroying that cell after the viruses have been, the viral particles have been assembled, so that now they can be released to the outside to find new cells where they can continue, this process of replication. So we're going to start now with lytic phages, and we're going to look at how the lytic phages can replicate. The first thing the lytic phage will do, and we already know these 5 essential steps, is they will look for a cell that has that particular receptor that can bind to the spike of that virus. If they find that cell that has a receptor, they will attach, like you see there. After attachment, the virus will enter, and in this case, we're seeing a mechanism of bacteriophage is injecting the genetic material into the cell. And once inside of the cell, then now that virus hijack the synthetic machinery of that cell and give instruction to that cell to just produce copies of that virus. Start to replicate the genome of the virus, start to express the genes, the viral genes, to produce the viral protein. And when those elements are produced, then now new viral particles can be assembled. After those viral particles are assembled, in the case of a lytic phage, then now that cell will be lysed, like you see here, and the viral protein will be released from that cell. For these lytic phages, this is a very quick replication, usually bacteria. The whole process can take, minutes. By the time, the viral particles are released. Let's take a look now at the second group of viruses that I mentioned, the temporary phage infections. And temporary phage means, it may react in different ways depending on the conditions. This is a temporary phage is a virus that has 2 options. It's a virus that could just produce a lytic infection, like the one that I just described, or depending on the conditions in the environment, the virus may go into a lysogenic infection in which the genome of the virus gets integrated in the chromosome of that cell. When the genome of the virus is integrated in the chromosome of the cell, we're going to call that structure, that viral DNA integrated, we're going to call that the prophage. So in that process now, in the lysogenic cycle, that bacteriophage genome is not expressed and is instead integrated into the bacteria's genome to form the prophage that I just mentioned. And since the bacteriophage genetic information is incorporated into the bacteria's genetic information as a prophage, now the bacteriophage is passed from generation to generation as the bacterium divides to form daughter cells. In this scenario, the daughter bacterial cells contain prophage, and this structure in which you see, the cells containing a prophage in the chromosome of the cell, we're going to call those cells lysogen. Now, lysogens, they can remain in the lysogenic cycle for many generations, but can switch to a lytic infection, all of the sudden, at any time. And this will happen through a process that we call induction. So through a process of induction, all of a sudden, the genome of that virus will be excised from the chromosome of that cell. And once excised, then now that virus will start to replicate, produce copies of that genome. The viral genes will be expressed, the viral proteins will be produced, and then now new viral particles can be assembled and finally released through a lytic process. That means a cell, they will be lysed to release the new viral progeny. And now the final group of bacteriophages that I mentioned to you in the initial slide is the filamentous phages. They're called filamentous phages because it's making reference to the shape of those viruses. They look like filaments. And that's the reason why we name this particular group of viruses like that. These viruses, like for example, in the case of the M13 phage, they can use, for example, the sex virus as the receptor to now inject the genetic material into the cell. The viruses start to replicate in that cell, start to assemble new viral particles, and then the assembly of new viral particles will happen at the cell membrane and cell wall. And now the new viral particles, they will be strewed through the membrane in a way that the cell will not be destroyed. So in the case of filamentous phages, the cells are not alive. The cells, they remain alive, and they keep shedding, virus out of that cell. The only thing that is going to happen here to the cell is that the cell is not going to be killed, but now that cell that is infected with this filamentous virus will grow more slowly. Now, what mechanism viruses have in order to fight these viral infections by bacteriophages? There are different mechanisms that what are the mechanisms that cells, bacteria, have to prevent infections from these bacteriophages? Well, you know that in order for the infection to happen, the first thing that needs to happen is the virus needs to find a receptor on the surface of the bacteria. Sometimes some bacteria like Staphylococcus aureus, what they will do is they will produce proteins on the surface of the cell that will mask the phage receptors, not allowing the virus to reach that receptor in order to attach. And if the first step of attachment is not happening, it's impossible for the virus to continue with that infection. Also, bacteria that produce capsules and slime layers, they may also, in occasions, they may prevent that virus from binding to the receptor because they could be just masking those receptors, not allowing the virus to get access to those receptors. Now, if the virus enters to the cell, what other mechanisms the bacteria could have to prevent this infection? Well, there is an important mechanism here, and it's the production of restriction enzymes. These are enzymes that cleave, that cut DNA into fragments at a specific nucleotide sequence. It could be at that specific sequence or near that specific sequence. And these are sequences that could be found in the genome of the virus, let's say in that DNA genome. And if the bacteria is producing those enzymes in the cell, and those enzymes, those restriction enzymes, are able to recognize that specific sequence in the genome of that bacteria, then now those restriction enzymes, they could cut that DNA in pieces. And if the DNA is destroyed, the genome of the virus is destroyed, then now that virus cannot replicate. Now, those sequences that the restriction enzyme recognized, they could also be present in the genome of the cell. They could be there in the genome of the cell. However, the reason why these restriction enzymes are not able to destroy their own, the genome of their own cell, is because the cell has modification enzymes that are in charge of methylating those sequences that can be targeted by the restriction enzymes. And when those sequences are methylated, then now the enzymes cannot cut that DNA. The only problem with these modification enzymes is that these modification enzymes, sometimes, they may also methylate the DNA of the virus. And if they modify the DNA of the virus, then now the restriction enzymes will not be able to cut that DNA. And now the virus will evade, those restriction enzymes and start the replication into the cell to produce viral particles. And then the final strategy that I want you to see in this presentation, and I will just speak about this briefly, is the CRISPR system, a system that has been identified in prokaryotic cells, in both bacteria and archaea. CRISPR is the acronym of clusters of regularly interspersed short palindromic repeats. Basically, this CRISPR system is a family of DNA sequences found in the genome of prokaryotic cells. These sequences are derived from DNA fragments of bacteriophages that had previously infected that prokaryotic cell. And now they are these sequences that have been kept there in the genome of the bacteria, these tiny pieces of the genome of previous infections caused by the virus. They are used to detect and destroy DNA from similar bacteriophages during subsequent infections. So basically, these sequences, they play a key role in the antiviral defense system of prokaryotic cells. It's kind of like an immune system that confers resistance to foreign genetic elements, like the gene present in the virus, right? It's kind of like an acquired immunity in bacteria, let's say. And basically, you can see here the process. Let's say, this bacteria here got infected with this virus. The genome of the virus is infected. That virus genome, maybe through the help of restriction enzymes, gets chopped in pieces. And now one of those pieces, the cell decides to keep one of those pieces of that DNA of the genome of the virus and insert that into this cassette that I mentioned to you, the CRISPR system, the CRISPR array that you see right here. Now, when that cell replicates, the future generation will always have in the genome that piece of the genome of that virus that one day tried to infect a previous generation. And the cell now is able to transcribe those fragments. And now that RNA that is produced from the transcription of those fragments of the DNA of previous viruses that try to infect will be on display on this Cas CRISPR RNA complex. And if a similar virus with that similar sequence all of a sudden try to infect this cell, and this could be a cell, a generation, many generations after the initial cell captured that sequence, right? If the virus with that sequence enters this cell now, this Cas CRISPR RNA complex will detect the presence of that sequence because it's going to be homologous and it's going to have a complementary interaction with that sequence. And after that is identified, then now this RNA harboring spacer sequence will help the Cas proteins recognize and cut the foreign DNA to destroy it before the virus can replicate into the cell. This is what we call the CRISPR system. Now let's go into animal viruses. And when I talk about animal viruses, these are viruses that can infect different animals. We are animals. I will focus my attention on human viruses because, again, this is a clinical microbiology, and by the end of the semester, we're going to be talking about different viral infections in humans. So I will stress probably processes that are related to human viruses here. Similar to bacteriophages, animal virus replication go through 5 essential steps. Remember, the first step that we discussed was attachment. Attachment, again, is the process that will guarantee the specificity of that virus to infect a particular tissue or a particular cell. Why? That virus will bring on the surface, a spike that will have, maybe a shape that allow that virus to infect this cell, because the receptor for that spike is present in this cell. But, for example, it may not be able to infect this other cell because the correct receptor is not on that cell. And this is what we call tropism, the ability of the virus to target a particular organism, a particular tissue in that organism, a particular cell in that organism. And that is the reason why maybe viruses that infect other animals, in many cases, are not able to infect us. But also, you know that there are viruses that infect animals that can be transmitted to us through this process that we call zoonosis. And in that case, what that is telling you is that those animals, they have cells that have that receptor for the virus. And in our body, our cells also have that receptor for that particular virus. And that's why they can be transmitted from these animals to us. Classical example, what we're seeing now with the coronavirus, with COVID-19, what has been proposed is that probably the origin of that virus is in a bat. The virus is able to replicate there because in the bat there are cells that have the receptor for that virus. And then now, at some point was transmitted to humans, and our respiratory tract system also has that receptor. And now we get infected with that virus. Like, for example, what happened with the HIV. HIV is a virus that can only infect humans. I mean, in the process of evolution, it has been suggested that probably comes from chimpanzee through a process of evolution that made that virus mutate, evolve, and now produce a viral particle that can specifically only infect humans. And HIV uses the CD4 molecule. The CD4 is called a cluster of differentiation 4. So CD4, that's why the molecule is called like that. Uses this CD4 molecule as the main receptor to attach to the surface of the target cells. And this molecule, the CD4 receptor, is expressed in cells that are part of the immune system. It's expressed on CD4 positive T lymphocytes. It is expressed on monocytes, on macrophages, and on dendritic cells. But that molecule is not expressed, let's say, for example, in the liver. And that is the reason why the virus doesn't infect the liver. is infecting cells that are part of the immune system, because those are the cells that have the receptor for the virus. But it's a virus that also needs the presence of co-receptors in order to enter the cell. But then again, these are the cells that have those receptors and co-receptors, and that's why the virus tropism is towards these cells that are part of the immune system. And hepatitis A, B, C, D, E, they infect the liver, because in the liver is where these viruses find the receptor for that. And rabies virus is able to infect the central nervous system, because it's in the central nervous system where the virus will find the receptor to infect those cells. And SARS coronavirus 2 infects the respiratory tract, because it's in that respiratory tract, upper and lower respiratory tract, where the virus will find the receptor for that virus. So this first step of attachment is a very specific step that will only happen if the virus has a spike that is able to interact with a specific receptor on a target cell. So after that attachment happens, then now the next step will be entry, similar to what we saw in bacteriophages, right? And that process of entry, can be divided in penetration and encoding. Depending on the structure of the virus, then the strategy that will be used to enter that cell. If we are talking about a virus that has that capsid with the genome inside, and then on top of that has an envelope with the spikes on that surface. Only in the case of envelope viruses could use this strategy of attaching to the surface of the cell, by interacting with the specific receptor. And then after that attachment happens, the envelope, right, the membrane that is covering the virus, the envelope of the virus will get very close to the cell membrane of the cell that is being infected. And then now those two membranes will fuse together and open now this space for the nucleocapsid of the virus to enter into the cell. And that nucleocapsid now will go into the process of encoding to release the viral genome, so that now replication of that virus can happen. Now, both envelope viruses and naked viruses can use the second strategy that I'm showing you here. They can enter through a process that we call endocytosis. And that means the virus will attach to the surface, and the attachment to that surface will trigger the process of endocytosis, in which the membrane of the cell start to engulf that viral particle. And now the virus gets into the cell, into this endosome, and the virus will be released from that endosome, will encode and release the viral genome to now start the process of replication. The third step in animal viruses, similar to bacteriophages, the process of synthesis. Now, the process of synthesis, or the strategy that the animal virus uses to synthesize copies of that genome and to start the process of expression of viral proteins, will depend, on the genome of the virus. And that genome of the virus and enzymes that the virus bring with them will dictate that strategy on how that virus will replicate inside of the cell. So let's take a look now first at viruses whose genome is a DNA. And remember that in the case of viruses, some viruses, they will use double-stranded DNA as a genome. Others will use single-stranded DNA. Let's take a look at both of them and how they can replicate. A virus that brings double-stranded DNA is usually a virus that can bring their own DNA polymerase to replicate that viral genome. Now, there are some double-stranded DNA viruses that may use the host DNA polymerase to replicate. So you can find both options there. That means after the virus enters the cell and the genome of the virus is released into the cell, some viruses, they may bring their own DNA polymerase and start the replication of that genome right away. Other viruses, they may bring their own, they may use the host DNA polymerase to replicate. When that genome is replicated and copies of that DNA viral genome are produced, then now, of course, the gene that the virus has there in that genome will have to be expressed. And those genes will be expressed usually with the help of the cell RNA polymerase transcribing those viral genes. Although, in some occasions, we may find viruses that bring their own viral RNA polymerase to do the process of transcription. And then finally, the process of translation is happening thanks to the cell. In this case, the virus is hijacking all those ribosomes in order to translate the viral messenger RNAs. It's using all the amino acids that are produced in the cell. This is completely, done by using the host cell machinery. Now, in the case of single-stranded DNA viruses, the single-stranded DNA viruses, they always use, to replicate the genome, they always use the host DNA polymerase. They don't bring their own DNA polymerase. So as soon as that virus is released into the cell, the genome of the virus, the DNA, is into the cell, then now the virus will use the host DNA polymerase to start the replication of that DNA, to produce copies of that viral genome. And then, of course, the virus also will use the cell RNA polymerase to transcribe the viral messenger RNA. And at the end, it's going to use the cell machinery for translation to produce the viral protein. So these single-stranded DNA viruses are viruses that will come into the cell ready to use all the machinery from that cell. They will use the host DNA polymerase to replicate the viral genome. They will use the RNA polymerase to do the transcription of the viral genomes. And they will use all the ribosomes and all the amino acids, transfer RNA, in order to translate those viral messenger RNAs to produce the viral proteins. Let's take a look now at RNA viruses. And here I will focus on single-stranded RNA viruses. Remember that I mentioned to you that also you may find double-stranded RNA viruses. I don't have time to discuss that particular strategy of replication today. I will focus right now only on single-stranded RNA viruses. And I mentioned at the very beginning that that single RNA genome of the virus could be positive or could be negative. What is the difference between those positive RNA genome viruses and negative RNA genome viruses? Well, here you have the difference. A positive RNA genome in a virus is a virus that as soon as that RNA enters into the cell, the RNA can be used directly as the messenger RNA to translate into viral proteins. And then this virus also at the same time is going to, it's going to translate that messenger RNA, and it's going to bring their own RNA polymerase to produce copies of the viral genome. Now, if we talk about a negative RNA virus, what that means is that when the RNA of this virus, the genome, is released into the cell, that genome cannot bind to a ribosome, cannot be translated. Before that happens, the complementary RNA strand has to be produced, and this will be the positive RNA that now can be translated into a protein. So the initial minus RNA, that is the genome, is not able to be translated into a protein. First, the genome has to be transcribed to produce the complementary RNA that now can work and add as a messenger RNA to be translated and produce the viral proteins. These viruses, in the case of these viruses, you will find some viruses that bring their own RNA polymerase, that is a viral RNA polymerase. There are others that will just use the cell RNA polymerase to replicate the genome. And then I mentioned that double-stranded RNA is a little bit more complicated in terms of the replication. I will not explain that today, but if you're interested in learning about the replication of double-stranded RNA viruses, I can explain that to you, during office hours or at another time. Then the final group that I want to describe here is a very particular group. It's called retroviruses. Right? Retroviruses include viruses like HIV. And these are viruses that bring, in the structure of the virus, they bring a genome that is positive RNA genome. A positive RNA genome, based on what I explained in the previous slide, is supposed to go directly into translation to produce a viral protein. It could add as a messenger RNA, but that is not what happened with retroviruses. Retroviruses, they bring with them very unique enzymes that allow that virus now to follow a different strategy for replication. One of the enzymes that these viruses bring is this enzyme called reverse transcriptase. The reverse transcriptase, what that enzyme will do is will do the opposite of what you know until now about the natural process of expression of gene expression. You know that the natural process is DNA being transcribed into RNA, and then that RNA being translated into proteins, right? Well, these viruses, they will do something very unique. They will now take that RNA and convert that RNA into the complementary single-stranded minus DNA. And that's why we're going to call this process reverse transcription, because it's the opposite to the transcription that you know until now. Transcription is using DNA as a template to produce the complementary RNA. Reverse transcription does the opposite. Reverse transcription is using RNA, positive RNA, as a template to now synthesize the single-stranded negative DNA strand. And this same enzyme, the reverse transcriptase, will now take that negative DNA strand and synthesize the complementary positive DNA strand to produce a double-stranded DNA. that double-stranded DNA now will travel to the nucleus of the cell. And in the nucleus, that DNA will be integrated in the cell genome. And that process of integration will happen also thanks to an enzyme that the virus bring with them. That enzyme is called the viral integrase. After that DNA of the virus is integrated there in the cell, depending on the cell that is being infected, that virus could go into a latent stage, and that prophage, similar to bacteriophages in the temperate group, that prophage could remain there silent for some time until the cell is activated and the virus can start to express, the viral proteins and start to produce new viral protein. Sometimes the virus infects a cell that is already activated, and as soon as that genome is incorporated in the genome of the cell, the viral genome is incorporated in the genome of the cell, it will start the expression of the viral genes. The viral protein will be produced, copies of the viral genome will be produced, and new viral particles will be assembled to release them from the cell. And the final steps that you have here are the process of assembly of the viral particles, and at the end, the release from those cells. Assembly is basically, taking those copies of the genomes and then now encapsulating them into the protein capsids that are assembled inside of the cell. If the virus is naked, the release of that virus, naked, non-enveloped, right here, naked viruses, the release of that virus will be by lysing that cell and releasing those naked viruses to the outside. Now, if the virus needs to acquire an envelope on top of that viral nucleocapsid, then one of the mechanisms that the virus could use is, assembling the nucleocapsid close to the cell membrane, inserting in that cell membrane the viral spikes, so that now the virus will bud out of that cell, acquiring that viral envelope that has the spikes on the surface. And we call that process budding. It's kind of like the inverse of endocytosis. In this case, would be an exocytosis, right? In order to acquire that viral envelope that has the viral spikes. So we're going to stop here now for a break, and I will continue in the second part of this class with approaches to cultivate and quantify animal viruses.